So if you've ever had that, that constant ache deep in your lower back, the kind that makes just bending down feel like a huge effort.
Oh, yeah. Or that morning stiffness.
Exactly. You are definitely not alone. Chronic low back pain is, well, it's actually the leading cause of disability worldwide. The economic impact is just staggering.
It really is.
So today, we're doing a deep dive into some really interesting new research. It's a study that offers maybe a completely fresh angle on this huge problem, looking right down at the cellular level.
That's right. We're stepping back a bit from the usual focus on, say, muscles or spine alignment for a moment.
Uh-huh.
And we're going to unpack this fascinating work looking at these things called, well, zombie cells. The technical term is senescent cells.
Zombie cells, okay.
Yeah, and their role in your spinal discs.
Huh.
The researchers are exploring this really intriguing idea that maybe we can clear out some of this biological clutter that could be driving the pain.
Zombie cells driving back pain. Wow, okay, that's definitely an angle. So this study, it highlights something pretty specific, right? Two compounds.
Yes, two main ones.
One from nature, surprisingly, and one synthetic. And they showed promise, in mice at least, in reducing back pain by actually targeting these senescent cells. And the natural one, get this, it's o-vanillin, comes from vanilla.
It's pretty amazing, isn't it? And what's really key here, I think, is that the potential shift this suggests. It's investigating a direct link between these seemingly inactive zombie cells and this very common problem of chronic low back pain.
You're right.
Because, as the study points out, a huge factor in this pain is the wear and tear, the degeneration of the intervertebral discs.
Those cushions between the vertebrae?
Exactly, the shock absorbers. They start to break down.
Okay, so the discs degenerate, maybe get inflamed, and that's where these zombie cells enter the picture. Can you tell us a bit more about what they actually are? Why are they causing trouble?
Sure. So senescent cells are basically cells that have stopped dividing. They've reached the end of their reproductive life, maybe due to damage or just age.
Okay.
Now, normally your body has ways to clear out old or damaged cells, a process called apoptosis or programmed cell death. But senescent cells, they resist that. They stick around, like zombies, hence the nickname.
Okay. They don't die when they're supposed to.
Precisely. But the real issue isn't just that they're lingering, it's what they do while they linger. They start spewing out this cocktail of inflammatory molecules. Collectively, it's known as SASP. That stands for senescence-associated secretory phenotype. Bit of a mouthful.
This ASP. Right. Yeah, sounds like something you wouldn't want hanging around in your spine. So this SASP creates a sort of like toxic soup in the discs.
That's a good way to put it. It creates this pro-inflammatory toxic environment. It's like this hidden low-level chaos happening constantly at the microscopic level.
And that makes things worse.
Absolutely. It worsens any existing tissue damage. Right. It irritates nerves. And it basically fuels the whole pain cycle. It keeps this low-grade inflammation simmering, driving the degeneration and the pain sensations.
Okay, I get it. So zombie cells stick around, pump out inflammatory SASP, and help keep chronic back pain going. So the study looked at senolytics as a way to fight back. What are those exactly?
Senolytics are a class of drugs or compounds designed specifically to find and eliminate these senescent cells selectively.
Like a targeted cleanup crew for cells.
Exactly, like a cellular spring cleaning. They often work by interfering with the survival mechanisms that these zombie cells rely on, kind of pushing them towards that self-destruct pathway they were avoiding.
While leaving healthy cells alone, ideally.
That's the goal, yes. To clear out the dysfunctional cells and therefore reduce all those harmful SASP secretions we just talked about.
Okay, makes sense. And the researchers tested this idea in mice, you said? How did that work?
Right. So they used a specific strain of mice. These mice were genetically engineered, so they were prone to developing disc degeneration much faster and earlier than normal mice. It helps mimic the kind of age-related changes we see in humans.
Okay.
And interestingly, these mice were also missing a gene called Spark. Spark is important for, well, think of it like maintaining the structural integrity of tissues, including the spinal discs.
So mice without Spark have weaker discs.
They're more susceptible to degeneration, yeah. It makes them a pretty good model for studying this kind of breakdown and seeing if a treatment can help.
Gotcha. So these mice, already prone to back issues, were treated with these senolytic compounds. Which ones did they focus on?
They looked primarily at two. The first, as we mentioned, was o-vanillin, that natural compound derived from vanilla.
Still find that amazing.
And the second was a synthetic molecule, RG7122.
Right. Let's talk about the vanilla one first. O-vanillin. I mean, beyond making things taste good, what's special about it biologically?
Well, that's what's so intriguing. It seems o-vanillin might have a kind of dual action. Research suggests it has anti-inflammatory properties itself.
Okay, so it could calm down that SASP effect directly.
Potentially, yes.
Yeah.
And, crucially, it also appears to have senolytic properties, meaning it can actually help eliminate the senescent cells.
Wow.
It's a great example of how, sometimes compounds in nature have these unexpected biological activities. Makes you wonder what else is out there.
Definitely. Now, what about the other one, RG71112? You said it's synthetic, and it was originally looked at for cancer. Does that raise any red flags, like safety concerns?
That's a really important question. Yes, RG7112, particularly at the higher doses sometimes used in cancer research, has been linked with toxicity.
Right.
So the researchers here were very careful. They used significantly lower concentrations in this back pain study. And the findings suggest that at these lower levels, it might still be effective at targeting senescent cells.
Especially when combined with the o-vanillin.
Exactly, especially in combination. It seems the two might work together synergistically, potentially allowing for lower, safer doses of the synthetic compound while still getting a good effect. It really highlights how crucial dosage and formulation are.
Okay, so they treated these susceptible mice with o-vanillin, RG7112, or both. What happened? What did they actually see?
Well, the results were quite promising, actually. First off, they saw physical improvements in the spinal discs themselves.
Really? Like actual structural changes?
Yeah. The discs in the mice that got the treatments looked more voluminous, healthier. It suggested some restoration of that cushioning effect that gets lost during degeneration.
That's huge. So it wasn't just about masking pain, the tissue itself looked better.
That's what the data suggests. And secondly, they saw changes in pain-related behaviors.
How do you measure that in mice?
They used standard tests, like measuring sensitivity to touch or to cold applied near the affected area. The treated mice showed less sensitivity.
Ah, suggesting less nerve irritation, less discomfort.
Exactly.
Yes, sir.
It indicates the treatments were having a measurable effect on their pain levels.
And the zombie cells, did the treatments actually get rid of them?
Yes, and that's really key. They measured the number of senescent cells in the spinal tissues,
Ah.
and they found a significant reduction in those senescent cells in the treated groups, particularly in the mice that got the combination of o-vanillin and RG7112.
Wow, okay.
So it strongly supports the idea that these compounds were acting as senolytics. They were clearing out those problematic cells.
That is pretty compelling. So the mice seemed better, their discs looked healthier, and the suspected culprits, the senescent cells, were reduced. That really does feel like a different approach, doesn't it?
It absolutely does. And there was more. They also looked at the spinal cord itself, in the regions that process pain signals. And even there, in the treated mice, they found fewer molecular signs associated with cellular aging and inflammation compared to the untreated mice.
So the benefits might even extend into the nervous system's handling of pain.
It suggests that, yeah, by tackling the problem at the source in the disc, you might positively influence how the whole pain signaling pathway functions.
Wow. So this idea, this vanilla compound with a punch, as they put it really does seem to have potential. It's still just remarkable thinking about vanilla.
It really is. It highlights the potential sitting in natural compounds. And, the fact that the synthetic RG7112 also showed effects at these lower doses, especially combined, suggests maybe a multi-target strategy could be effective down the line.
So thinking bigger picture then, what does this research really imply for how we might treat chronic back pain in the future? It feels like moving beyond just, pain pills and physical therapy.
Exactly. I think this research points towards a potential paradigm shift. Instead of just managing the symptoms, the pain, the inflammation, we might be able to actually target one of the root causes.
These senescent cells driving the disc degeneration.
Precisely. The idea that we could potentially reverse or at least significantly slow down the degenerative process itself by clearing these cells, that's incredibly exciting. It offers a whole new type of hope, really.
It definitely does. A different kind of hope than just coping.
Oh, yeah.
We have to mention the caveat, right? This was a study in mice.
Absolutely critical point, yes.
Yeah.
While these results are very encouraging and provide a strong scientific basis to move forward, we can't just assume it'll work the same way in humans.
The jump from mouse to human is always a big one.
It is. We absolutely need human clinical trials. Those are essential to figure out first, is it safe for people? Is it effective? What's the right dose? How would you even deliver it effectively to the spinal discs? What are the long-term effects?
Right. Lots of questions still.
Many questions. That clinical translation phase is the crucial next step.
Okay. But even with those hurdles ahead, the basic concept here, tackling chronic back pain by clearing out these zombie cells, it's just fascinating. It really expands how we might think about what's going on in this incredibly common condition.
It does. And it makes us think differently about aging too, doesn't it? It suggests aging isn't just a passive decline, but involves active cellular processes like senescence
Yeah.
that contribute to disease.
Yeah.
And if we can identify and target those processes like clearing senescent cells, maybe it has implications not just for back pain, but potentially for other age-related issues too. Things like arthritis or cardiovascular problems, where these cells are also being studied.
So, wrapping this up, the main takeaway from our deep dive today is this really intriguing link. Senescent zombie cells, spinal disc breakdown, and the potential for senolytics, especially something like o-vanillin from vanilla, to actually target a root cause of chronic back pain.
That's the core idea, yes.
It offers, well, at least a glimmer of real hope for the millions dealing with this, suggesting a future where treatment might go beyond just symptom management.
Indeed. And it leaves us with a thought, doesn't it? Could something seemingly so simple, derived from vanilla of all things, hold a key to alleviating such a widespread and complex problem? And maybe more broadly, what other conditions tied to aging might be driven by these lingering zombie cells? It certainly gives us a new lens for thinking about cellular aging and our overall health.